everyone. So as Maya said, I'm Jessica Bullington, and I'm a first-year master's student in the Department of Oceanography at University of Hawaii, and I'm working with Dr. Craig Nelson. And my thesis will be using modeling to predict pathogen risk in the Ellison Canal. And I just wanted to orient you where the Ellison Canal is. So up in the top right, you can see Hanauma Bay. And to the left of the Waialua or Diamond Head crater is Ellison Canal. And it's fed by the Manoa, Pololo, and Makiki streams. And it was built in the 1920s to drain the Waikiki wetlands. So why do we care about pathogens in the Ellison Canal? Well, bacterial pathogens cause infections, and they can be fatal. So some of the pathogens that we see here are listed on the left, and the one that we're going to be focusing on today is Vibrio vulnificus. And in 2006, Oliver Johnson is thought to have died from a Vibrio vulnificus infection. And these pathogens are in the water column. They're free living. There's a picture of them up on the left. And we're going to be thinking about them as plankton. And so they show them as plankton from SpongeBob because it's a little more fun. And there are certain conditions where we might see a lot of plankton in the water. And so you might get an infection from direct exposure. If you have an open wound, it'll be infected. And then another way is that it will get into our seafood, and we'll eat it in crabs and other seafood. So we really want to know where and when they're going to be in the water. So thinking about their distribution, I wanted to go through an exercise of our own distribution on, remember that the globe is a sphere, but we're looking at it flat for convenience. So this is just one way to think about human populations through light pollution. And you can kind of get a feel for maybe what environmental conditions change our distribution around the globe. So there are very few people living around the poles. Maybe it's too cold for most people. There are very few people in the deserts, so maybe it's too hot. Maybe depending on like how good the soil and other things about the environment to dictate our distribution. We can do the same thing like this for biology. So this is a satellite map from chlorophyll pigments, which are light harvesting pigments in primary producers. So the way you can think about that is this is the distribution of the base of the food web in the oceans. And so you can predict these primary producers also based on environmental conditions, like how much nutrients are in the water, how much light they're experiencing, temperature, other things like this. So bringing it back down to Hawaii, our environment is the Ellison Canal, and the distribution that we're interested in is of Vibrio. So some of the things that we measure in the environment are temperature, oxygen, organics, which is food for Vibrio, and light, things like this. And we might measure the distribution or the diversity of Vibrio species. The environmental conditions are really easy to measure as compared to the biological, which are relatively harder to measure. And if we can understand the relationship between the environment and the biological life, we can use a model to predict the biology, just measuring the environmental conditions. So another way to think about that with an analogy is that the physical chemical environment is like a roadmap, and the response is road vehicle traffic. And then the model is our best guess of what the rules of the road are. So in creating this model, this predictive model, we have two components, lab experiments and field sampling. And lab experiments inform the model, so that's how we build it. And the field sampling, getting an idea of what's actually happening in nature, is what's validating the model. So how well does the model actually work? And on the right, this is actually the blue line is the transect of sampling days. So I'm just starting in my master's, so I've done a couple days of field sampling, but I have not started the lab experiments yet. But we're going to talk about what I potentially will be doing. So we're going to be growing Vibrio in different conditions. One of the conditions will be different salinities. And then we measure how fast their population is growing. And from this, we can get sort of optimal salinity that they really like to grow at. And salinity is basically a measure of how salty the water is. So from the streams, we're getting a lot of fresh water, which is really low salinity. And the ocean, we're getting salt water washing in, and we have this gradient of salinity. So that kind of data is going to be informing our numerical model. And it might look, this is a plume model, so how murky the water is, but it's actually not measured, it's modeled. So with mathematics, we can kind of guess what's happening while we go out every time and measure. So this was a storm event, and we can see that the water kind of flushes the Ellison, and that water is going harbor, but it's also going, close to Waikiki. So it's influencing the beach and our like fish harvesting. And as we grade out that space, as you can see on the right, and for each cell, we apply this method. And it's sort of like inputs minus outputs. So you have immigration minus emigration, so that's really just water movement, and births minus deaths. So that's getting at this like population growth curves that we were doing in the lab experiments. So the other part of this, once we have that model that we think is approximating these distributions, then we want to get a feel for how well it's actually working. And so we do these field sampling throughout the canal. And we have, so we're on a boat, and we put sensors in the water, and we're also taking water for to like actually quantify the Vibrio cells. How many are there in this water from the same depth. So we have one way of sort of measuring both the environmental and the biology at the same time. So a big aspect of this is not only the spatial change, but also the temporal change. So how is this going to be affected by rainstorms and tides changing, sea level changing? And we have a really dynamic environment, which is why estuaries are really interesting to study. We also have global climate change that is going to be affecting this interaction. So we know maybe what's happening with temperature and sea level. There's sort of these changes that are happening globally, and we have very little knowledge of what's happening with what might happen with pathogens. So we don't want our future to look like this image on the right, which is why these modeling approaches are really important to give us an idea of what we might be faced with in the future. I don't have any results for you yet. I'm just getting started. Hopefully next year I'll come back and have some results for y'all. I just wanted to thank, these are all of the principal investigators on this project. And you can visit these 2 sites, get more information about these projects. Thank you. All right. So, aloha everybody. My name is Devon DeBevoise, and I'm going to be talking about how climate change affects seedlings, in specific, the effect of temperature, CO2, and soil moisture on the performance of native and non-native woody tree seedlings here in Hawaii. So first of all, what is climate change? Well, climate change is just long-term changes in the global weather, which can include changes in temperature, wind, and rainfall. And these can be caused by gases such as CO2. Now, climate change impacts both land and sea, as seen in this picture. We see that over on towards the ocean, we have changes in the pH, in the overall chemistry, and circulation changing. But we also see it in the land. And so when we increase CO2 emissions, eventually we're going to end up with higher carbon dioxide or higher CO2 concentrations, higher temperatures, and changes in rainfall. And in this example, especially here in Hawaii, is we're seeing an increase in drought. Another cause is extreme events that are changing, such as storms. So we're seeing an increase in frequency in these kinds of events as well. Now, all of these affect the terrestrial habitat. And in my specific area, it's affecting the plants. But the main question is, but how will these plants respond to these interactions of climate change, to CO2, to temperature, to soil moisture, which I'm talking about as well as drought? And so this is the driving question behind my research, is the interaction and how they are going to affect the plants. So how are these plants going to respond to these climatic changes that are co-occurring? Well, we have a lot of knowledge on single variable responses. So we know what happens in presence of CO2 and in presence of just temperature. And general findings can conclude that plants use CO2, they take it in from the atmosphere in order to provide themselves with nutrients. CO2 also can influence how open the stomata are or how the stomatal conductance is occurring. So that can influence drought tolerance. And we might see that warming can typically benefit plants, as seen here in this example. So as we see in this little graph, as the temperature increases, this hypothetical plant, we see an increase where the yellow arrow is pointing. We see an increase in plant growth as we increase temperature. Now, all of this seems like a pretty good deal. All right, but we're just looking at them individually, individual variables. But it's where the conflicting expectations occur when the variables interact, because climate change isn't just CO2 changing. It isn't just temperature changing. It's an interaction of multiple variables that are occurring together. So if we're taking our same example, as the stomata are closing, that stomatal conductance is being reduced, plants can overheat due to the fact that transpiration can occur because the stomata are closed. And same thing, if the stomata are closed, no matter how much CO2 is in the atmosphere, if these stomata are closed, there's no way that CO2 is getting in, so these plants are going to start to suffer from carbon starvation. And so a lot of the research that's been done has been in cooler regions. And this is a picture showing examples that have been collected that talk about climate change, and they are all scientific papers from cooler regions. The issue that we have is that different areas respond to climate in different ways. So if we take an increase in CO2 and an increase in temperature, and we look at, say, the temperate region where temperatures are typically cooler than, say, here in Hawaii, we might see that we see an increase in growth due to the fact that the ambient temperatures in that region, the surrounding region's temperatures, are lower or are below the optimum level. So, and we also might see that precipitation might increase as well, especially in higher latitudes. So we would typically see a positive trend. We'd see the plant growth increase as temperature increased. Now, if we look at the opposite area in the tropics, such as here in Hawaii, we're not going to see that same reaction. We're going to see a decrease in growth due to the fact that the ambient temperatures are already above the optimum level. It's already warm. So as we increase temperature, we're also going to be increasing the amount of drought that's occurring. So we're on the opposite side of our little graph. So we're seeing as temperature is increasing, plant growth rate is declining. So it's extremely important, especially when drought is mixed in. Growth can be severely diminished. Now, what we're looking for and what we need is to be looking at the different types of experiments that are out there. And a lot of the controversy and contradictions that have been occurring in research may have come from the limited nature of the experiments that have been performed. So what we need in scientific research is experiments that encompass a range of variability of these climate change variables. So we need to see a range of temperatures, a range of CO2. But unfortunately, experiments like these, the ones that we want and the ones that we need, can quickly become unmanageable without the help of automated growth systems. So these automated growth systems or automated growth chambers can test these multiple interacting variables. And I have two examples here. The first one is the WIWAM, which is the weighing, imaging, and watering machines for scientific research. And as you saw there, these are automated robotic systems that will weigh and record all of the information for you. It's amazing. And then we have another example called the phenoscope. This is another type of automated growth chamber that is out currently on the market. Now, these sound pretty cool, right? These are just two of the examples. The problem is the price. These can range from $100,000 to $200,000 per chamber, right? And so the price of these also prohibitively restrict the amount of research that can occur. And so what we want to do is to overcome this challenge. And so here at the University of Hawaii, we have our PIs on the grant is Dr. Camila Mora and Dr. Zachary Trimble. They have developed something called the Intelligent Plant Growing System, or the IPS Growth Chamber, to overcome this technical challenge, to overcome this financial challenge. So with our chambers that we have on campus, we're able to alter CO2 levels, vary the soil moisture level. So we'll be able to change how much water is available to our plants within the chambers and alter the temperature within the chamber. So each individual chamber will be set at a specific temperature, a specific CO2 level, and then a range of water treatment water availability. And we have 10 working chambers. So we've got these built, we've got these ready to go. And so these white containers that you see in the middle of the photo, those are actually where the plants will be housed in. So those are our containers the plants will be growing in, inside these chambers. So once we seal these chambers, the lights will go on for 12 hours and 12 hours off. And then they will be completely automated and we'll be able to observe and record the data without actually having to go into the chambers. Now, the objectives of our experiment is, the first one is to assess plant growth and survival. And we're going to do that by observing those three gradients, okay, the three variables. We want to observe the gradient. So of CO2, temperature, and soil moisture, water availability. Our second one is to estimate overall plant responses to projected climatic changes. And we're going to do this by combining our experimental research with existing climatic projections. And also to identify potential mechanisms for experimental patterns in plant growth. And we're going to do that by looking at the link between growth, survival, and the overall plant performance.
Now, climate change is expected to alter the biodiversity, which will in turn affect species interactions. So we want to look at how native and invasive species react and respond to these changing environments. So our first one is our native, it's our ohia. And we've chosen this well-researched at University of Hawaii, and it's of its cultural significance here on the islands, and it's also undergoing that massive mortality due to the invasion of the invasive fungal disease. And so our research showing how they will respond to ongoing projected climate change may be able to aid in the overall restoration process. And our invasive species that we've chosen is the strawberry guava, and we've chosen this because it is recognized as one of Hawaii's worst invasive species out there. And it invades a lot of native Hawaiian forests and replaces a lot of native plants and also destroys a lot of habitats for native birds and insects as well. So being able to compare how invasives and climate change will again help aid in the restoration process. And by observing and by measuring a suite of morphological, anatomical, and physiological traits, along with seedling fitness, we aim to shed some more light on the controversy and how plants will respond to climate change. And some of our example of measurements in our morphology, we're looking at leaf thickness, anatomy might be vessel diameter, physiology might be stomatal conductance, and fitness like biomass and survival. Thank you very much, and I'd like to thank my PIs, Dr. Casey Barton and Camilo, and the Sea Grant for allowing me to be here as well. So thank you all for listening to me.
All right. Hello, my name is Jordan Mason. I'm a first-year master's student at the University of Hawaii. I'm part of the Department of Geology and Geophysics.
I'm Lucas Ellison. I'm a first-year PhD student. I'm his partner in the same lab in geology and geophysics.
And then our principal investigator is Craig Glenn. So we're going to talk to you about cesspools and the sea. Is sewage polluting the North Shore? Okay. So first, I wanted to give you a little background. So this is the island of Oahu, where we are. We're down here somewhere. Over here. So this is showing OSDS density map. OSDS stands for On-Site Sewage Disposal Systems. So all private homes have these, and that's where the waste is stored from your house. So the main thing to note here is all of the hotspot areas in Waianae, Waialua, Kahuku, and Kahalu are all coastal areas. And there's approximately 14,000 OSDS on the island. And then the majority of these are cesspools. So our main focus area is Waialua Bay, which is on the North Shore. And in this area, it's a high priority area because there are 1,900 on-site sewage disposal systems within or adjacent to Waialua Bay. And 1,400 of these, or 75%, are cesspools. And as you can see, it's very close to the coastal water, so it's an important thing. Okay.
So here's another map of our study area. If you look on the right map, you see all these red dots. Every single one of those is a cesspool. So there's a huge amount of cesspools in this area. And there are other OSDS types. There's aerobic and soil treatment. But cesspools are the real area of concern because if you look at the diagram, in case you don't know what a cesspool actually is, it's a pit of brick. It's designed to trap the solids of your sewage at the bottom and the liquids leak out. The idea is the liquids will leak out and eventually leach their way into the water table. And this is by design. They're going to be naturally filtered, get rid of any harmful pathogens and nutrients. However, the big issue here is distance between the cesspool and the water table has to be great enough for this filtration to happen. And if you're in a coast, the water table is going to be very high and it's almost not gonna ever gonna filter out because the water table is right there. Another big concern is if you're at a coast and there's a high tide, this may flood and everything will become flushed out of the cesspool and into the surrounding groundwater and even the floodwaters. So Now I'm going to talk about nutrient loading. So nutrients, they're essential to life. They're in pretty much everything, in the soil, rainwater, than all of us. However, excess nutrients in coastal waters can lead to large algal blooms, like as an example here on the west coast of Maui, huge invasive algal bloom. And they often come from anthropogenic sources, like excess amounts come from anthropogenic sources, I should say, like agriculture, fertilizers, pesticides, herbicides, all get into the water, make their way to the coast, and then human waste from cesspools and septic tanks and other OSDS types.
So one of the main topics of our group is submarine groundwater discharge. So we have to be able, one of the main things we have to do is we have to know where groundwater is entering into the coastal water. So, and this is called groundwater discharge. So what it is, it's a mix of fresh water, which would fall from precipitation, and then it would infiltrate into the soil, into the sediment, come down, enter into the freshwater lens, and then it would also mix with salt water that comes from this side, enters in, and they mix together, creating brackish water. And then this brackish water is semi-saline, and so it's less dense than the saline water of the normal seawater. So when this enters into the coast and it enters into the coastal waters, it will float on top of the saline water. And this is very important for us because it helps us locate where groundwater enters into the coastal water.
So now we're going to get into our actual methods and what we're going to be doing. We're just starting this our first year. So our purpose is to determine the hydraulic and geochemical connectivity between terrestrial sources of anthropogenic contamination and waters of Waialua Bay. So basically determine the connection between OSDS and the coastal waters, see if their water's getting from the OSDS to the coast. And our hypothesis is that nutrient loading from this OSDS and agriculture into surface waters and groundwater will impact the water quality in these coastal waters in Waialua Bay.
So the first thing we have to do, as I said, is we have to be able to find where water enters into the coast. And then once we know where it's coming from, we can figure out what it's made of or what's inside the water. So we do this by remote sensing with drones. So as you can see in this picture, I'm there playing with the drone, setting up our FLIR camera that we put onto the drone, and then we fly it up into the air. And then we fly along the coastline, and it'll take infrared images of the very first millimeter of the coastal water. And so what's important about this is since the groundwater that's entering in from submarine groundwater discharge is less dense, it's going to float. And then also because the groundwater hasn't been exposed to the sun, so it comes out a lot colder. So that makes it easy to determine where groundwater is entering and what's actual seawater based on infrared imagery. So in this image, even though this is an image from a plane that took infrared imagery, we'll be using drones. The red and hot area is normal seawater. Anything that's not red, so anything that ranges from a color from orange to blue, has been mixed with groundwater. So it's where groundwater has entered. And it's hard to see, but along the entire coastline from about here, in here, there's some blue, and then blue inside the harbor as well. So all these locations are where groundwater is entering into the coastal water. And then, yeah, okay.
Okay, the next thing we'll do is we'll be doing water sampling. So we're going to get samples of water from streams, groundwater wells, ocean water, as well as directly from cesspools so we can see what sewage is made out of. And we'll be measuring for nutrients such as carbon, phosphorus, nitrogen, nitrate, phosphate, all that. And then carbon and nitrogen stable isotopes. Coastal radon survey will be done to determine, that lets you know at the ocean where groundwater is coming out. So groundwater coming out from, as submarine groundwater discharge, will have radon in it because of the way the rocks decay. And we'll be able to tell if there's a high amount of radon, that's where groundwater is coming out. We'll do microbial tracers to see if there's any fecal bacteria in the ocean waters that suggests they're coming from OSDS wastewater. We'll be doing a water quality analysis at each of these sampling locations. We'll be getting the pH, temperature, salinity, and dissolved oxygen. The salinity and temperature, as you saw earlier on the last slide, the temperature you can use to see where the groundwater is because it's colder. And the salinity also, groundwater won't be as salty as seawater. It'll be a little bit salty because it's brackish mixing in there, but it'll still be less salty than ocean water. And then as an example of how this works with identifying it, this graph is a little complicated, but I'll just explain what you need to know. This stable isotopes of nitrate in water. So you got NO3 here, And then you see the N value of the NO3 is here and the oxygen value is here. If the N value of the sample is high and the oxygen value is low, if it fits in here, then you know this probably came from manure or septic waste. That's just an example. However, with doing this, you can't always tell where it came from because there's this thing called a denitrification line here. So what happens here is you'll get your values for it, but as water's in the ground, it'll start to be processed by microbes living there, and that will change the amount of N. It'll increase it because it breaks down the nitrate and releases it as nitrogen gas. So you may have a value here right in the middle of this square, but there's a possibility that it started here and then has been groundwater for a long time and moved like this way slowly. So we'll be doing that. We'll be using the other samples and parameters we're measuring for to clarify where it came from for sure. But that's just one example.
Okay, and then some of our final methods that we'll also be using to understand the hydrology of Waialua Bay is we'll be doing groundwater modeling. We'll start with a more basic model, and then as we collect more data and stream heights, stream gauging, when we do from our stream gauges, where streams are losing and gaining water, we'll add more to the model. And then hopefully by using the model, it'll help us predict where we can get the best samples for our water quality, like where we know water is exiting and going into the coast, or where we know it's coming into streams or leaving streams. So the model will hopefully help us understand and get more accurate, or let us find areas to get more accurate measurements. And then we'll also be doing stream gauging, as I said, to get stream heights of all the major systems in the area. And then we'll also get the flow rates, and this will all be used to help build our groundwater model. And then the final thing we'll also do is seepage runs, where we'll go up the major river systems of Waialua Bay and transect all the way up them and find out where the stream's losing water or gaining water. If it's above the water table, it'll lose water because then the water will fall into the, so if you look at this diagram, the water's above, the stream's above, so it loses water into the water table. Once the stream goes below the water table, water enters into the stream, so it's a gaining stream. And so knowing where it's losing and gaining can help us figure out where nutrients are leaving the water or entering the groundwater. And it'll, again, just help us further our groundwater model and make it more complete. So as we said, this is our first year, and we've just scratched the surface in this first semester of what we're supposed to do. We haven't done much. We hope to pick it up more in these next couple months and do all of what we just told you. And then next year, hopefully have some results to present to you. So thank you for your time. And are there any questions for everybody?
So how will the speakers just stand here? All right, so we have about 5 minutes for questions. Does anyone have a question? Yes, sir.
Vibrio, first speaker. What's its background occurrence? In the ground, in the water, where is it?
So we find it in the water column and also in the sediments. And it's actually a natural bacteria for Hawaii, which is why it's not monitored by a lot of the state-run water quality monitoring, because they say it's natural, so maybe it's not as harmful. Yeah, and we actually see it more often during rains, but not really intense rains where it would flush all the water out. But sort of these lighter rains where you're getting a lot of organic nutrients put into the water.
How pathogenic is it? Why is it? Why are you focusing on that one agent?
It's really harmful if you have like if you're older or if you're sick and you're like predisposed to this infection being really harmful. But most people that are healthy in good condition probably won't have this infection become a problem for their health. And I'd say that we're focusing on this one specifically because it's understudied, because it's been told that it's natural, so maybe it's not as harmful.
This question, so are you gonna test, like, say, under what condition that Vibrio has the highest density in the water? Is that-
Yeah.
So people can.
So high density, low density, wherever that is, what conditions will make that a problem for human health? Higher densities being more of a problem.
Anyone else have any questions? How long will it take you to complete this program?
So my master's is going to be 2 years. So hopefully I can finish all of that by the end of 2 years. And there's been some ground work already been done for the modeling, but they don't have as many natural samples. So being able to really assess whether that model is well predicting what's happening in the natural environment is where I'm sort of stepping in.
I had a connection from different so for the plants, I think I may have missed that. Are you starting them from seed, or they will be seedlings before?
We've actually begun growing them, so we start from seed, and then they need to get to a certain height before we can transplant them.
And then within each of the chambers, will you have native and the invasive?
Yes. So we're going to be, each chamber can hold 50 containers or 50 pots. So the Okia and strawberry guava will be intermixed with one another, so we can determine how they respond in the same environment. It would be a different experiment altogether if they were completely separate experiments, and then we might not be able to tie the strings together between the two. Thank you.
And for the cesspools, I was wondering, do you expect to see that there's groundwater coming through and affecting all of the coastline? I guess you should flip that question. Do you think there's any place around the island where groundwater is not going to be found along the coastline?
No, I'm pretty sure along most coastlines at portions of it, it's always discharging into it. There are certain areas where it's discharging a lot more.
Yeah, like
And those are where you can actually see it.
it's like, yeah,
I think it's always discharging into the coastal water. Though.
yeah, and it's kind of deceptive how much actually discharges. Globally, it discharges more than rivers into the ocean,
Yeah.
so it's pretty much everywhere.
Quantifying how much groundwater is getting into the coastal water, that's a lot harder to do.
And how much of that is sewage?
Yeah.
Right?
Yeah.
Then on your first slide, you guys had a hot spot on the windward side. Do you know where that was at?
Oh, yeah.
Yeah.
Where is that one? Do you know?
For
The one.
this one, right? I'm not.
Little further.
This one?
South,
South.
yeah. Keep going.
Or south. The other side.
Oh, that one.
Yeah.
Good question. Are you gonna test the impact in marine life?
So that's what a lot, people have done this project beforehand and we haven't necessarily gone far enough. We hope to see what the actual effect is on marine life. But
Yeah, we have a plan for a reef health study in our grant, but that would be in association with biology group. Wouldn't necessarily be us too much.
Last question.
I had a question for Devon. Potentially, I missed it, but you were saying that the other machines cost either $100,000, $200,000, and you talked about the one that you're using currently in the lab. Is that cheaper? Your invention, how much would that cost?
So the geography department and the engineering department collaborated together, and they built the entire thing. So we cut down the cost to about $1,000 across chamber.
wow.
So that's why we were able to do 10. And sooner, and then depending on the research we get out of these, we would like to expand.